This is the title of my thesis, and so this is exactly what I did during the last 5 years in my PhD. And before diving into exactly what I did, I would like maybe to explain a bit more what is the topic. So what does in-field phenotyping means and why it's important. Phenotyping actually refers to measuring relevant traits of the plants in the field. And the reason why this is important are mainly three. The first one is for resources optimization. So knowing where plants are growing faster can help us deciding where we want to use more or less water or more or less fertilization. The second reason is for breeding purposes, which refers to selecting which variety we want to use in our field. And for example, this is the teosinte plant, which is the ancestor of the modern corn. So what happened is that actually breeding help us into making this plant growing bigger with more kernels that we can feed more people. And as last thing, phenotyping help us understand the interactions between the plant and the environment. There are several elements that can change plants, for example, different temperatures, scarcity of water, the presence of pests. And while we could monitor all of this information, it's actually way easier to just look at the plants, and when these appearance change, we can actually intervene into the field and try to solve the issues. There are several tasks in computer vision that help us enabling phenotyping, so measuring these traits starting from one RGB image. The first one is called semantic segmentation and aims at classifying each pixel in the image as either crop or weed or soil. In this case, the crops are depicted in green and the weeds in red. Then the second task is called plant instance, which takes all of these pixels that we classified as either crop or weed and actually try to separate them into single plants that are here depicted in different colors. And one could go one step further and separate each one of these plant instances into their component leaves. And once we have the leaves, we can actually start performing the real phenotyping tasks, so measuring their length, their width, or their curvature, for example. Now, most of these tasks are nowadays learned through neural networks. And when we say learn, we usually refer to supervised learning, which starts with an image and a label. This label is a semantic one where we can see still the crop and weed and the soil in black. And usually what happens is that a researcher or some assistants need to label all of these images, so select each of the pixels and define which class they belong to, so that a neural network can process the image and predict an output. The prediction in the beginning is actually very confused because the network doesn't know yet what we want from it. But if we show several labels for different images, for thousands of images actually, the neural network can then learn what we want from it, so that at the end, our prediction is exactly matching the label that we have. However, for most of my PhD, I actually work with unsupervised learning, which basically means that we are not using the label. And one could ask, why do we care about unsupervised, as we were seeing that the supervised version was working quite nicely. And the main reason is that labeling is really hard, while collecting unlabeled images is relatively easy. If we look at these two semantic labels, for example, we can see that crops and weeds do not always look the same, because obviously we have different fields with different varieties, different crop species, and often they have very thin structure that are hard to labels. And looking at different publicly available datasets, we discovered that labeling just one of such images requires an average of 2.5 hours. And overall, we also need to do this several times to perform a quality control, as this is exactly what we use for the network to learn. So we want them to be perfect. It's clear that when we need to do this for different tasks, different fields, and for thousands of images, this does not scale very well. And this is why we care about unsupervised, because we tackle the bottleneck of data labeling and remove the labels. However, the main problem here is how do we perform the learning if we do not have the label? And there are several techniques that have been explored in the literature. However, for most of my PhD, I mainly focused on computing what are called pseudo labels. In this case, the pseudo label is a vegetation mask. So we use the green color from the image to define which pixels belong to vegetation in green and which pixels belong to soil in black. And this can be done in a geometric way, so just using the colors without using any network and without using any researcher. So it's less time consuming and obviously less expensive. And we can use this pseudo label exactly as we were using the label before, so that the network learn this task and can perform basically a vegetation segmentation. If we look at these, however, we are not getting exactly the same result that we had before because we are missing, for example, the width information. So using just the color does not give us enough information to differentiate between crop and weeds. This is why most of the time we're using pseudo labels. We still need to show some real labels to close the gap. In this case, however, we are using less labels, so less time and less money to just let the network learn how to distinguish between crop and weed once it knows already how to distinguish vegetation. The last thing that I want to explain before diving into the technical contributions is actually what we used to enable unsupervised learning, and this is domain knowledge, which the definition is the knowledge about workflow, configurations, and constraints of the environment in which we operate. And the reason why this is important is that this knowledge actually enable us to reduce the complexity of the problem, and so to actually produce pseudo labels and to enable the unsupervised part of learning. During my PhD, I made several contributions to this topic, starting from approaches that are actually meant to be used combined with supervised learning, so trying to improve their performance and reducing the reliance on labels. Then I developed two approaches that are actually performing semantic and instance segmentation without any label. And at the end, I also moved from the 2D to the 3D domain, trying to tackle the task of leaf instance segmentation and actually trade estimation, reducing the amount of manual labels that is needed. However, for time constraints during this presentation, we will mainly focus on the unsupervised approaches. So in this first part, I'm going to explain how we perform the semantic segmentation in an unsupervised way. And the domain knowledge that I used is called spatial arrangement because, first of all, the semantic task I want to remember you is aiming at classifying each pixel as either crop, weed, or soil. And as I said, the knowledge that we want to use is the spatial arrangement. Most of the fields in agriculture look like this. So we have a clear row structure for the plants. And this means that we can actually use this information to already have a prior about where crops and weeds should be. However, most of the time, the platform that it's moving onto the field does not have the whole image, but just one part of it. So what was in the red box. And this is why I'm going to show you the approach on one single image. So let's say that this is the image that our platform gets from the field. The first thing that we do is actually compute the vegetation mask. So use the green color to select where is vegetation and where there is just soil. And then we detect the main line. So we want to detect the crop row. And this is done with a computer vision algorithm that selects the line that overlaps the most with the vegetation. The second thing that we need to do is actually be able to detect other lines. For example, this one, which is propagated from previous images. If you remember how the robot was moving onto the field, actually we have one line detected at every image, which means that we can actually, knowing our position, we can use the previous lines and propagate them onto the current image. So the line, the light blue line that we just saw is actually coming from a previous image. And I think that the video is not starting here. It's coming from a previous image. And we projected it into the current one. So this is where we get from, we will get to. And once we have the lines, we can actually label, we can actually assign all the vegetation that is on this line to the crop class. So we can color them in green as before. Now, most of the approaches would label everything else as weed. However, we know that using lines is not perfect And most of all, these areas around our lines may contain some crops that were not perfectly aligned. This is why we decide to do not label anything else that is in these areas and just assign the weight class to everything that is outside of these areas. So as you can see, we put in red only elements that are completely outside of these two regions. At this point, we actually performed our unsupervised semantic segmentation, and we can compute some metrics to check how good they are. And the metric that we use for semantic segmentation is called intersection over union, and is defined as the area of overlap over the area of union, which means that if this is the area that we want to detect as crop, and this is the area that we actually detect as crop, the green part is the overlap, while the red part is the union. So this means that this is a value between 0 when the two areas have 0 overlap to 1 when the two areas are perfectly matching. And we can usually express it as percentage between 0 and 100%. So these are actually the results of our pseudo labels. We can see on the x-axis 4 different datasets that we tested on. On the y-axis, our metric, so the higher the better. And our approach in green outperforms all of the other methods on all the datasets. The other methods that we use were two unsupervised learning approaches, so based on neural networks. There are the blue and the red one, while the yellow approach is another geometric approach very similar to ours, but it was not capable of detecting the second line or a second crop row in the same image. And as I said before, these are our pseudo labels, which means that we actually want to train on them. And what happens when we train on these labels? We first need to understand what is actually changing. And the first thing is that we will predict soil, crop, or weed also for the objects that we did not label in the first instance. And what we hope for is that all the other examples that we give the network of crops, weed, and soil enables to learn some features so that the new prediction are actually better than the previous one. And if we compare the results of the pseudo labels against the network trained on these labels, we can see that both our approach and the second best approach, that was the yellow one, are actually gaining some performance. But what I think is the most important thing is the comparison between our approach and the manual labels, which is the purple bar, and which represents basically training the network, same condition, but on labels that are manually produced, so by a researcher. And we can see that the difference is just 2% below. Basically means that the gap is very, very small, even if we did not use any manual labeling. So we actually ask ourselves, can we do something better? Can we reduce ulteriorly this gap? And what we can notice from this label that we produce is that there is way more green than red, which is obviously normal as most of our crops are bigger because we do not want weeds to be there. But these also create an unbalanced distributions while training. And this means that it makes the network uncertain about the weeds. And before diving into how we want to exploit this uncertainty and this unbalanced distribution, we should know what does it mean for the network in terms of uncertainty, and basically what the network doesn't see, the network is not able to predict. So if you remember in the first instance, we only had at a certain point vegetation and soil, so the network was never predicting weeds because we were never seeing them. But also what the network sees rarely makes the network unsure because the network does not have enough examples to understand how that specific class or that specific element should look like. And uncertainty for neural networks can be separated into two big parts. The first one is called epistemic, and it captures the lack of knowledge, which means that the network did not see enough data. And the second one is the aleatoric, which means that there is ambiguity in the data. So the network saw two very similar elements that actually have different classes, for example. However, we can compute, given our pseudo labels, both of these uncertainty and estimate them, answering basically two main questions, which are how often does the network see this class for the epistemic, and how often does the network recognize this class for the aleatoric. And if we can use this question to compute the uncertainty, we can actually use it to change the predictions. So let's see how we're actually using it on this specific image. I use this image because, as we can see, there are a lot of weeds in the bottom left corner that are in line with the crop. And this is why the network that we trained on our pseudo labels actually makes a bit of a mess in that area because it's seeing a row, so it thinks that everything below should be a crop. However, if we look at the uncertainty computed as we said before, where purple means low uncertainty and green, yellow means high uncertainty, we can see that all of the weeds are actually very uncertain for the network. And this is because the class is the one that it's not seeing enough. So we can use that information to actually change the prediction of these pixels from crop to weed and correct our predictions. In doing so, we actually gain a 7% IOU on average over the different datasets. And most of all, for the case that we've seen before, the difference between training with our pseudo labels and the manual labels is now only 0.5%. So we gained a 1.5% of performance. Now we can see some qualitative results over different datasets. This is an onion dataset, which is very hard. As we can see, the onions that are in green have a very thin structure. And we can see now our approach on the left and another baseline on the right. And we can see that we are not error-free. There are some small mistakes that we are still committing because one weed was in the line of the crops. And this obviously it's something that we cannot fix. However, we can also see that all of these crops, we are leaving them unlabeled, so we are committing less mistakes than if we were just labeling everything. On the other side, we can see that the baseline is actually committing more mistakes, even close to ours. So for example, those two weeds are actually labeled as crops. And the inability to detect more crop rows actually creates a bit of a mess on the right part where everything is labeled as weed, even if there are a lot of crops there. So as a summary of this first part of the presentation, I show you how we can exploit the knowledge of the field arrangement to produce pseudo labels for the semantic segmentation task, and how training on our pseudo labels actually yield a performance that is just 2% lower using manual labels while having zero manual annotation, and how to exploit the imbalance between crop and weeds to actually reduce the gap to even 0.5%. Now, in the second part of this presentation, I'm going to move on to the plant instance segmentation task. And the task, as I said before, is to assign a unique ID to each pixel that is part of the vegetation, trying to distinguish between different plant instances. And how difficult can it be if we have already a semantic segmentation? And actually, the answer is that it depends on the growth stage. For example, for this task, any geometric approach would perform a very good result because, as we can see, all the plants are perfectly separated one from the other. However, the more the plants are growing and overlapping, the harder the task is. For example, even as a human, I would be a really hard time trying to figure it out where each plant starts and ends in this image. So the knowledge that we try to use in order to perform an unsupervised plant instance segmentation is the crop morphology. In the case of sugar beets, which is the plant that is depicted here, but also in the case of broccoli and cauliflowers and many other plants, the plants are actually growing in a radial way, which means that in every stage, from the small one to the bigger ones, all plants can be enclosed in a square. So the aspect ratio of this instance should be roughly 1. So the height and width should be kind of the same. And this is actually the information that we want to use to separate the single plants. I'm going to now show you how we perform it using this image on the left, where if we just use a connected component, so if we just do a semantic segmentation and then try to separate them, this is what we would get. So the white instance would be all one because, as we can see, it's all completely connected. The first thing that we do is try to find a way to separate this plant. And in order to do so, we want to look for edges. So we use an edge detector on this instance, which we detect as a problematic instance, as obviously the height and width are not 1. So this means that we actually have more plants that we want to detect. So the edge detector actually gives us this result, where in white we can see the edges and in black everything else. we can see that the edges are not perfect. We are not getting just the separation between plants, but also between leaves, and often the stems that are a little bit wider, they are also depicted here. However, if we cut them out from the previous instance, this is already a little bit easier for us to differentiate the single instances. And what we do is actually take all of these cut out boundaries and make them a little bit larger. So then now we have a clear separation of each component. At this point, we use our information about the desired aspect ratio to define how many plants we are looking for. So again, we compute the height and the width, and we compute the number of plants, that in this case was 4, and we assign the 4 biggest component as center of our new plants. They are now depicted in pink, green, light blue, and purple. All the other components cannot be a new plant. It means that they need to be assigned to one of these 4, and we do so using the closest one. And we do exactly the same for all the pixels that we just cut out, but that are actually part of the vegetation. At this point, we actually already have a plant instance segmentation, but we can see that we only use basically distance in the image directions. And this means that, for example, this leaf over here, which is pink, is assigned to the pink plant because it's the closest one, but it's not connected to it. So we actually do one more step in which we do a cleanup, and we enforce that each plant should be fully connected. And in this way, we actually can fix this problem and let the leaf be connected to the green plant. At this point, as before, we also need to have an evaluation metric, and the common evaluation metric for semantic and instant segmentation together is called panoptic quality, which is based on the IOU, so in the intersection over union that I explained before, but also counts the match positives and negatives between the instances that we detect and the real instances. And in order to understand what every of these terms is, I'm going to show an example. Let's see that this is our ground truth. So these are two plants that we want to detect. And in gray, I'm now showing our predictions. So two plants that we are actually predicting with our method. And in this example, we actually have all 3 terms that are on the equation. A TP is a true positive, which means that our prediction is matching a real plant. So there is a good overlap between what we think is a plant and what's a real plant. Then we can see a FP, which is a false positive. So it's a plant that we think is there. We are predicting as a plant, but it's not actually a real plant. So it's a false prediction, basically. And then we have the false negatives, which are plants that we should predict, but we are not predicting. So it's a missed prediction. In this way, the panoptic quality is higher when we have matching predictions. We are not seeing plants when there are no plants. We are not missing plants. And the plants that we are predicting are actually matching the most with the real ground truth. And we show here the performance of different networks trained on our instances. So on the x-axis, we see 4 different architectures, so 4 different networks. On the y-axis, the metric of the panoptic quality. And actually, the results in blue are training the network on manual labels, so using 100% of human annotations. And this is the average over different datasets. And then in red, we show what happens if we remove half of the manual annotations and we substitute them with our labels. So we can see that actually the performance most of the time is increasing. So our labels are actually improving the performance in most cases. And moreover, this is happening while reducing the labeling time of more than 1,000 hours on average. So we are not compromising on the performance, but we are reducing the time required for labeling and so also the cost of labeling. The second experiment that we did on these 3 different datasets on the x-axis was actually trying to see how we can improve existing methods. So using our approach after a first plant instance segmentation, we used 3 different methods, a graph-based one, so totally geometric in red, and then 2 visual language model that are like the common approach nowadays for plant instance segmentation. And if we use our approach on top of the results, we can see the improvement that we get in green. And we can see that we improve over all the different scenarios, all the different datasets. And most of all, these gain us an average of almost 20% more panoptic quality over the different data sets and approaches. So I'm going to show you again a qualitative results. What we can see on the left is the results of a baseline. We can see that it still has a hard time separating plants that are connected to each other, but using our post-processing approach, we actually are able to separate each single instance quite nicely. And as a summary of this for the second part of the presentation, I show you how we exploited our knowledge of the crop morphology to actually identify the number of plant instances and then the real instances. How we were able to reduce to half the need for labels without compromising on the performance, and actually how we can improve plant instances over different crops and starting with different methods over different scenarios. As I said before, was only the part 3 and part 4 of my PhD contributions. I have actually contributed into improving the performance of supervised methods in the part 1 and 2, and I was actually able to improve the 3D leaf intersegmentation and then perform a real leaf trait estimation in part 5 and 6. And this is basically the contributions that I did over time and reducing the amount of labels over all the different scenarios. And we actually generated a 3D single leaf trait dataset to improve the leaf trait estimation. And this was everything that I did during my PhD, and I thank you for your attention, and I'm open to any question.